Hello, my name is Benjamin Thomas, and I'm here to present the project that I've worked on in the fall and in the spring, improving gene editing techniques with CRISPR Cas9 in the yellow fever mosquito, Aedes aegypti, Diptera chelicity. And so the reason why Aedes aegypti is a species of interest in these experiments is because they can vector Zika virus or dengue virus, and even in some cases, yellow fever virus or chikungunya. And so millions of people yearly are infected with these viruses, and it can cause high morbidity, the state of being diseased, or high mortality, the state of being dead, in tropic and subtropic regions, also known as these high-risk regions. And so these high-risk regions come up with control methods, whether it be chemical or biological, to combat these pests. And so an example of chemical control could be going from the use of insecticides or insect growth regulators. And for biological control, there could be the use of using small fish, for instance, in standing bodies of water, where mosquitoes like to lay. And so but both have their downsides. As with chemical control, mosquitoes can develop resistance, and with biological control, a bigger predator, say in the area, could come by and eat these fish. And so I like to present a novel control method that is in development and currently in research, gene drive. And so if we recall Mendelian inheritance, we remember that classical genetics, that a gene could be subjected to probability through each generation, and so it would be harder to get whatever gene you would want through. And so with gene drive, that is the self-continuation or propagation of a selected gene or trait. And so it is faster than Mendelian inheritance. And so with gene drive, imagine if we can genetically engineer a mosquito that is unable to vector these viruses, then that'll be very beneficial for these people that are in high-risk areas. And so how can gene drive be perfected, or better yet, be implemented with the use of CRISPR Cas9, a gene editing tool? CRISPR Cas9 causes these double-stranded breaks that are tailored to a sequence. And Cas9 is a protein, a homing endonuclease, that is guided by a guide RNA that has this target sequence. And so this guide RNA essentially drags Cas9 to that piece of DNA and causes a double-stranded break. And so the question leads me to, what happens once this DNA is broken on both strands? Different DNA repair pathways compete with each other to repair this DNA. And so the most dominant repair pathway with fixing this double-stranded break is non-homologous end joining, NHEJ, and is known to be accurate in some cases, and in other cases result in small deletions or insertions. And so another closely related pathway, alternative non-homologous end joining, is known to be more error-prone and have larger deletions or insertions. And so these two pathways are known to be error-prone. And with gene drive, we would want a repair pathway that is not error-prone and yet perfect. And so homology-directed repair, or HR, has accurate recombination of this DNA. And so how can we test for this, and how can we understand the difference between error-prone versus non-error-prone repair? And so I made two plasmids, non-homologous end joining 2.0 and non-homologous end joining 2.1. Both have a promoter of PUB, which stands for polyubiquitin. Polyubiquitin leads to the expression of everything downstream of it, so the GFP or the FF loop. FF loop stands for firefly luciferase, and that is a protein that leads to the expression, a gene that leads to the expression of a protein that helps give off this luminescence. GFP, green fluorescent protein, was also put into this plasmid to essentially reorient the cut sites that these sgRNA Cas9 combinations will be cutting at. And so the only difference between these two plasmids is the cut site sequence. And so there's 4 base pairs between the cut sites of 2.0 and 5 base pairs between the cut sites of 2.1. Both plasmids were made to be out of frame, and thus giving no luminescence. And so what difference, and why does that matter? So this one base pair difference leads to differences in frameshift mutations. A frameshift mutation is an insertion or deletion of nucleotides that causes either the introduction of a stop codon or maybe the re-expression of a protein. And so in this case, when 4 base pairs are deleted, it will lead to this plasmid being essentially higher in luminescence and end frame. And then when 2.1 is deleted, 5 base pairs, then I'll lead to it still being out of frame and lower or no change in luminescence. And so how do I create these plasmids? Through the use of restriction enzymes, I was able to open up a construct and then to place it in the cut sites and then ligate these plasmids. They were then transformed into E. coli bacteria colonies, and they were selected for on an ampicillin-resistant plate. Once they were plated, they were then went through a colony screening to essentially make sure that we have a positive for this plasmid versus a false positive. And so the PCR, the primers chosen for this PCR mirrored these cut sites. And so on the gel that was ran, these cut sites were judged based off their size, and whichever one had the size of the expected product, they were then sent off for sequencing. And once the sequencing results came back, and we had the expected sequence, I then digested each of the plasmids with 1 sgRNA or 2 sgRNAs. And so 2.0, for example, was digested with 1 sgRNA Cas9 to get 1 cut site, 2 sgRNA Cas9 to get 2 cut sites, and the same thing was done with 2.1 to give us 6 varying conditions between the plasmids. And so once they were digested and cut at the cut sites, they were then transfected into Aedes aegypti embryonic cells. And so transfection is the process of bringing a plasmid or genetic material into a eukaryotic cell. In our case, that will be the Aedes aegypti embryonic cells, also known as A20s. And so a transfection reagent was used on these plasmids and essentially made a complex that essentially got brought into these Aedes aegypti cells to form an endosome, and that endosome would break down, leading to the plasmid being introduced into the nucleus. And so once this transfection was completed, Rhinella, another plasmid that can produce this luminescence, was also transfected into these Aedes aegypti cells. Why? For the dual luciferase assay that will be ran after. Dual luciferase assay essentially stands for a 2 light reporter test. And dual luciferase assays are ran to typically measure these biological activity. And so here's a depiction of the well plate for a clearer understanding of what was exactly ran and transfected. And so each 0 essentially means that there are 200,000 A20 cells that were transfected with 2.1 or 2.0, with the cut sites being cut or not, and Rhinella. And so once the dual luciferase assay was ran through a SpectraMax, a machine that can read these luminescent signals, results were produced via graph pad to make multiple ANOVA graphs. And so we see that with our results that the plasmid was not essentially out of frame, thus I need to come back and recreate it. But we do see, however, that with 1 sgRNA Cas9 combination, that cut, we see that there is more error-prone repair than with 2. And so we see on this graph that there was no non-error-prone repair, so no perfect repair was displayed. With 2.1, we see that 2.1 with no sgRNAs and Cas9s cutting, we see that there's 0 value, and that would indicate that it's a perfectly made plasmid. However, with 1 sgRNA cutting, we see that there was more error-prone repair. We see that with 2 sgRNA Cas9s cutting, we see that there is less error-prone repair. And when we see both of them side by side in tandem, we notice that the Y value for the Firefly over Rhinella led to higher values for 2.1. And so maybe 2.1 is a better suited plasmid. And so, judging off these results, we can say that there was no case of non-error-prone repair. Because as you recall, 2.0 will lead to higher luminescence if there was perfect repair. And if there was 2.1, perfect repair would be lower luminescence or the same amount of luminescence displayed. And so, we can also say that 2.1 is a better suited plasmid for this dual luciferase assay. And so I would need to come back and recreate 2.0, as 2.0 with no cut sites being cut should not have been presenting luminescence. And so 1 sgRNA and Cas9 led to more error-prone repair as seen with these results, and further experiments are needed as if you recall, non-homologous end joining in some cases can be perfect and thus can be used for gene drive. And so, and also I can come back in these experiments and of course send the cells off for sequencing as we can see what happened to these cells on a molecular level. And so with this research, it was mixed significant results and insignificant results. However, this research can increase our understanding of DNA repair in Aedes aegypti, as my results kind of steered me in another direction for future projects. This project can also provide insight into improving gene editing techniques in Aedes aegypti and other medically important vectors such as like ticks, and in some cases like termites. And this research also has the potential to reduce the public health burden of morbidity and mortality around the world. And thank you for your time, and I'd like to thank Dr. Edelman, my PI, and my research mentor, Justin Overcash, and basically everybody else in my lab, as they all contributed to my doing of this project. Thank you.
Can you go back to the graph which you were showing and explain to us what are we actually seeing in the graph? Like the spots and everything. Yeah.
So if I go back to the concept slide, so what we're looking for with perfect, we're trying to distinguish perfect versus non-perfect repair, right? Because we want perfect repair for gene drive research. And so 2.0 and 2.1 were essentially trying to measure these instances of perfect versus imperfect repair. And so if 2.1 was to display this perfect repair, it would have higher luminescence, and so a high value on this graph. And if this was to display perfect repair, then it would have lower luminescence or the same amount produced. And so if I go back to the graph, all right, 2.0 did not display any perfect repair, only error-prone repair, because it was relatively the same as if it wasn't cut at all. And so with 2.1, as you recall, it's not supposed to have higher luminescence if it was perfectly repaired. And it does have higher luminescence, thus indicating that it was error-prone repair. So it repaired not perfectly.
Okay.
Did that help?
So the numbers on the x-axis, what do those mean again? Like the 2.1 or the 2.0?
So 2.1 plus 1, essentially, 2.1 would be the plasmid, right? And then 1 would indicate that one of the cut sites were cut.
Okay.
And then 2 would be 2.
So if these values are higher, then that means the DNA repair did not work perfectly. Right? But it varies between 2.0 and 2.1.
Correct.
Okay. One another question. And so people are usually using CRISPR Cas9, to create mutations and things like that. And we want the cell to kind of have a messed up DNA repair happening at that region so that you get the mutation. But your aim is to have, allow the DNA repair to happen properly, right? So how does that help you in your aim? I'm a little confused with the aim of the gene drive aim, yeah.
Because with gene drive, if you wanted to put this in say an environment, a pest, say in this case mosquitoes, if we have to error-prone repair with this CRISPR Cas9, then it could lead to unexpected mutations, and then that could be potentially dangerous for the environment. And so for gene drive to be approved and to be into the nature, it would be feasible to have a perfect repair. And so we can have an expected mutation happening each generation.
Okay.
Do these mutations kill the mosquitoes or just make them infertile, or?
There could be mutations that can make them infertile. It can make them unable to vector these diseases. Mutations could go along the lines of making them less susceptible for these diseases also.
Okay.
So eventually you are making mutations, but you don't want the mechanism for the DNA repair to be error prone. Like you want that to be a very predictable mutation so that every generation you kind of get the same kind of heritable mutation so that the gene drive can happen, right?
Exactly.
So application-wise, is this something you would do in conjunction with like pesticides as well as the biological agents, or is this something to replace both of those since they both have downsides like you mentioned?
I think in the future this could be a use for replacing these control methods, because both control methods that I mentioned, like the chemical and the biological, they have like cost to it and labor. And then pesticides will obviously damage the environment in some cases. And so I think gene drive could be a cleaner method if perfected, but it's a lot of ifs there.
Mm-hmm.
Big